So fantastic. So today I will talk about living computers. My name is Ewelina Kurtys. I have PhD in neuroscience, and after my research, I moved to industry and I started to work on technology. Actually, I became really fascinated with artificial intelligence, which is actually a lot inspired by neuroscience, especially this project. So this is FinalSpark. This FinalSpark is currently a group of 6 people. It's a company founded in 2014 by Fred Jordan and Martin Kuter, which are 2 Swiss entrepreneurs. And they had a dream to build thinking machine. And they tried different approaches. They realized that silicon AI is very expensive. And this is why in 2018, they started to consider living neurons as alternative to silicon for the superior artificial intelligence model. And yeah, why it doesn't go? So which problem we are solving? So we believe that currently AI has a scalability problem, which we don't see today. It's not obvious yet, because a lot of AI which we are using today, especially, for example, large language models, they are quite cheap because they are still in the adoption phase. So actually companies who make them, they are still losing money. We know that OpenAI lost probably something like 30 billion US dollars. So it's actually really expensive to do AI, and this cost will increase exponentially. That's what is expected because models are better, which means better models mean more energy usage, and we will use them more. So that means all this energy cost will be increasing. And we believe that in some time it will become a problem. So we have actually 2 possible ways. Either we find alternative energy sources, or we will find alternative computing to solve this issue. And what FinalSpark believes is that we can replace digital processor with bioprocessor, which will be built from living neurons. So the same as we have in our brains. And just for illustration, exactly what we do. So we use the building blocks. So we don't try to reproduce human brain, of course. That's too complicated, and that's also not the goal. We want to build artificial structure, but build from the same building blocks, living neurons. And currently we have a small prototype, which you can see here on the picture, this round structure. This is called organoid. It has around 10,000 neurons each and 0.5 millimeter diameter. And we put them on electrodes. And usually they are just 8 electrodes. And that's our basic system, our prototype of the future computer. And why we use living neurons? So first, because we know it works for sure, because the best proof for that is that we can talk today is because our neurons are processing information. The second reason is what I also mentioned before, the scalability problem of current silicon system, and living neurons are 1 million times more energy efficient. So we could solve the problem of the current digital systems, which are using too much energy, and because it's scalable. So actually it's relatively easy to produce huge amount of neurons in the lab currently with the current methods which we know. And these are our organoids. So, you know, biology is much more messy, much more unstructured than engineering. These are organoids under electron microscopy. So you can see this is very messy structure. And this is a little bit of biology, of course. So living neurons has to be always stored in right temperature, 37 degrees Celsius in this way. Also, they need to have medium, which is special liquid with all the nutrients and oxygen, everything what living cells need to survive. So we always have to remember about this biological part. And then we connect everything. We connect our neurons to electrodes, and they are connected to digital systems. So this is what engineers know very well, and they feel comfortable in this. And actually, we can control living neurons from the computer. So the way how we do experiments is that we write code in Python programming language, and this code is automatically executed on the neurons. And the basic idea is that we can programmatically send them electrical and chemical signals, because this is how neurons are communicating, and we can also read the response. And this is also example, I have it on the next slide. So this is the setup in our lab. So you can see again, mess is always like that with biology. That is very complex system. Is very it seems to be chaotic always. And this is the readout. So what we can read from neurons. This should be the V. No, it's going. Oh no, sorry. Yes, it's not moving. Okay. But on our website, you can see it's moving. FinalSpark.com, you can see the same picture actually. And this is the typical signal which we get from living neurons. So this is what we analyze also. So we send them first input, chemical or electrical signal, and we measure this kind of output, which is electrical signal. And we try to make sense between those input and output. And this is the next slide. This is our team. So as I mentioned, it's just a 6 people, very small team, a mixture of engineering, biology, mathematics. But I think it's a perfect mixture for this type of project. And then what is special about our lab, that our lab is totally available remotely. So as I mentioned before, doing experiments in our lab means writing code in Python. So that means it doesn't have to be done actually in our lab, because the Python code can be written anywhere in the world. And indeed, researchers from all over the world are working with us. There are university researchers, also companies have some crazy ideas, and they can write code in Python, which is automatically executed in the lab in Switzerland. So the same way as our scientists are doing, and they collect data, they can write papers and do all the work without ever entering our lab. And actually, what is biggest advantage here, that we allow people to do this really breakthrough research on biocomputing without the necessity to run wet lab, which is a lot of pain, especially for engineers, because most of the people who work with us are actually technical, because there's a lot of data, so they feel comfortable with this and analyzing big amounts of data. And for these people, yeah, of course, it will be a huge barrier to build a wet lab. And especially such an advanced wet lab, because our neurons can live for quite long, around 3 months, which is really long. Seems to be short, but actually for the standards, it's really long. And yes, so we make this research possible for many who couldn't do them if they had to build a lab themselves. And this is a little bit about our achievements. So yes, we have Neuroplatform, which is our lab, live 24 hours per day, 7 days per week, automatic experiments, so you can run huge amounts of experiments. We also have 9 universities working with us. We chose them out of 34 candidates, and these are our scientific partnerships. But we also have increasing number of commercial clients who rent the lab on the subscription basis. So you can rent basically FinalSpark Lab for 1 month, and you can run experiments on your own. We managed to store 1 bit of information in our system. We also published the paper, which you can see the screenshot. This paper is it's the top 1% of papers read in Frontiers in the journal when it was published. And we also have 2 patent families. So again, this is the slide I already showed you. If you would like to get in touch, of course, you are very welcome. You can scan the QR code. You can also find easily me on LinkedIn, and you can very easily find our website, finalspark.com, and you can send us a message. Very easy to get in touch with us. We have a lot of forms, a newsletter, and Discord channel. So there are many ways to get in touch with us, either directly by asking questions or joining our community. So highly encourage you to this. If something comes to your mind, some idea, or maybe you would like to try experiments on living neurons, then feel free to get in touch. And thank you so much for your attention.
Thank you, Ewelina.
Thank you. I will stop sharing.
Yeah.
Moment. Okay, no.
Yes. Okay, Brett. Stage is yours.
Great. Well, hello, everybody. Pleasure to see you this evening, morning, afternoon, wherever you happen to be in the world. I'm Brett. I sit as both Chief Scientific Officer and Chief Operating Officer at Cortical Labs. So I get to wear a lot of different hats. Today, I thought I'd wear more of a scientific hat and dive into a little bit about how these systems work as part of this really awesome workshop. So Where did Cortical Labs start? Essentially, we started with the question, is it actually possible to harness biological living neurons outside the body for computation, intelligence, and learning? And this led us to a more straightforward question, which is, can we get cells in a dish to play Pong? This was back in 2019, and there hadn't been any breakthrough real-time closed-loop work in this way that had actually demonstrated robustly that neural cultures could be directed in a goal-directed manner. And we combined a bunch of different techniques and were able to actually bring about a system that showed this learning. And while this was a great scientific question, this is obviously Neurofounders. So we actually had to have a company idea behind that. And the question became, how did we turn science into a platform? And we did that essentially by having 4 pillars: software, computational neuroscience, hardware, and wetware. And I'm going to run through these quickly with you now. So the computational neuroscience, I'm going to go pretty quick on because we don't have hours. But essentially, there's a lot of really interesting features that biological neurons have in the way that they work. They're highly scalable. They can be highly specific in terms of their computational properties that we can elicit through a range of synthetic biology methods. They display what's called at least third-order complexity. In other words, they can hold up to at least 3 dynamic states at once, potentially more, and operate on them in an analog scale. If you compare that to silicon, which is only a first-order complexity, it can have one state, and in that manner, a binary state. This is obviously a huge advantage. They can also learn with significantly less data. We actually published a paper that got a bit of attention showing that these cells could learn quicker than a range of reinforcement learning algorithms. They're adaptive. They're able to use far less energy than AI, as we've just heard about. And they can be developed with a range of features with real-time learning. They deal well with chaotic and noisy data, and they don't show classic limitations that AI systems often do, like catastrophic forgetting. Now, the core of, if I was to summarize the core of what you need to test computational properties, is this idea of embodied networks. Now, an embodied neural network is essentially a closed loop feedback system where you actually stimulate the cells, you measure the response to that stimulation, and then you provide sensory feedback on how this actually alters the world that the cells are embodied in. Now, this is really obvious for us as humans. If we act or move, we get immediate feedback. For cells, it's harder, right? So ideally, it has to be high resolution, real time, and physiologically relevant. And that's where our software comes in. And so we've recently released the CL API, and I put in quotes here, the CUDA of biological computing, because that was what someone was kind enough to refer to it as on a review online, which is nice. And essentially, this is an API and a developer kit that actually allows rapid developments of new environments, even by non-experts. And this is a key part of what unlocks applications for our platform. And far higher reliability than any system that exists on the market for this particular purpose. And that's, I think, really shows the care that we've put into our system to make sure that it's able to engage in the types of explorations that us and our users want to do. On top of that, we also have our wetware. And we do our own world-class biology. For example, we develop new neural cell types. These here are fully specified hippocampal cells, which have been a thorn in the side of people doing hippocampal work for over 10 years now, because they've been unable to generate some of the key subtypes. We've actually done that here with all the subtypes required, and some of these organoids are still going. When we did this work, they went up to about 450 days, but they're still alive, some of them, so they're now going on beyond 500, 550 days. The interesting thing, though, is a lot of people talk about organoids, but organoids is just one option. So you can actually create a whole range of different cell types, monolayers like we did for our classic Pong work, organoids like we've been doing recently, or really interestingly, these microstructures that are highly controllable. And we just heard about how biology is messy and inconsistent. This makes the biology reproducible to a very significant extent. And there's pros and cons for each. So monolayers are very easy to set up, but they have little structure and they have little reproducibility, very hard to control. Lots of activity, can have really dynamic connections, but you're really hoping the biology works out. Organoids, we've heard a lot about them, so I won't go into huge depth, but they're complex, 3D, they can represent physiology well, but highly variable. For example, here we have 3 different organoids all next to each other, and you can see the activity footprint is totally different. So they're still good for some things, but if you want controllable and reproducible, you can go to these custom structures where each of these wells has a set number of neurons, and you can actually see here the connections between each of these wells. And what this does is moves it away from physiology, but into the realm of controllability. And so, for example, you can build these highly reproducible, highly structured patterns of activity across all the organoids. And if you do, sorry, all the organoids, all the cell cultures. So if you do 10 of these cultures, you're going to get 10 patterns that look almost identical to this. All right, the final pillar, hardware. Like all great startups, we also started out of a box from our local hardware store. Australians will recognize Bunnings. And we basically had to build out two parts. We had to build out a perfusion circuit to keep the cells alive, and we had to build out an MEA system to actually interface with the cells. And that led us over a few years of development to this pre-production model of what we call the CL1. And this basically integrates a housing unit with our CPU and FPGA that allows us to actually interpret and run the low-level code that the CL API directs. It has an MEA interface where a range of different MEA can be used with 60 electrodes. And we have a perfusion circuit system as well as gas and all the other support you would need. And then we've scaled this. So we actually have our own labs with 120 units running now. Many of these are also available to be accessed over the Cortical Cloud. And this actually opens the technology to the world. So we have our own cloud-based system that you can log on, access, and use. And there's been people who indeed have been using it. We started first, of course, and like all scientists, we aimed to replicate our Pong work, which fortunately we're able to replicate our old work that we did on the off-the-shelf hardware with our own hardware, and even get some modest improvements there, which was nice. As well as doing other tests, like seeing could cells learn Morse code, could cells learn things like handwritten digits. I'll note here quite a nice performance gain with the modular units over organoids and monolayers. And other people have been building too. So some people might have seen we had some cultures doing Doom done by students out of a hackathon, although it was actually Sean Cole who was outside of this hackathon who did this particular version, which was quite nice. And we've had more high throughput automation studies. For example, here, this was some work done with a European user, and there will be a paper released on this in the very near term, done in collaboration with them, where they actually ran through this high throughput study over about 3 weeks, iterating through a lot of encoding parameters to actually see that you can get a biological agent to get better at chasing basically an odor gradient, this bright green circle. So you can kind of see at the start, the agent wanders around, can't do much. By the end, they can perform quite well. And we found, we could get up to a 3.3 times improvement over baseline. So I'll wrap up there and sort of note that we have the one platform, the CL1, but there are 3 opportunities. So if you want to buy a unit for your lab, you can. If you want to just contact us and get us to do a project for you because you have a question you want to answer, you can do that too. And if you want to access it yourself and you can do a bit of Python coding, although our system is fully compatible with all the Gen AI coding assistants because it's all fully documented. So even if you don't code, you could take a run at the Cortical Cloud. That's available as well. And I think I'll end on this note that a lot of what we're talking about with biocomputing sounds a lot like sci-fi. And I've had that a lot. People said sounds like sci-fi. But science fiction stops and science begins when we start working on the problem. And what our focus for Cortical Labs has been is to allow you to work on the problem with us. So if you do want to do that, visit the website, send an email, reach out. We'd love to work with you on making this science fiction into a science. Thanks, everyone.
Thank you. Amazing. So if I understand correctly, we can soon vibe code on Neurons. Or nice. Yeah, no, I think it's good to start with that. Also with the sci-fi comments. To a lot of people, it sounds like sci-fi, but there is already customers. You both have a list of customers. So who are those customers and what are they using this technology for right now?
So yeah, we can both say. So our
Feel free, please go first.
Yeah, our customers are usually some technical geeks for sure, all of them, no matter where they work. It's still quite a technical challenge to work with living neurons because you cannot like analyze data. You cannot use it like a cloud computing today, but that's the goal in the future. So yes, they are usually, most of them are engineers. and technical fields, and they try to find patterns in the data. Let's say, I would say this in such a way.
And do you have any like use case study which you found most interesting so far?
Actually, we have a lot of ideas. Our objective is learning in vitro. So we want to be able to control living neurons and make them learn some tasks in vitro. That's our objective. But our users are free to choose ideas. We help them only to assess if they are feasible. For example, recently, one of our users published a paper. This is first publication of our collaborators about trying to encode Blair letters into living neurons. These letters data were coming from tactile sensing from the robots. So very interesting, with an idea that maybe one day robot can have living neurons to analyze this. Of course, this was scientific exploration, it's not a product idea. And it was just to show if there is anything possible to be done, because at this stage we are kind of trying to see if anything can be done on neurons. We cannot really do useful things yet. But yes, I think that's interesting, and you can find it on our website, exactly in our blog, the summary of this paper and a reference to the full text. I think this is interesting example, and there are a lot of projects which are ongoing, and of course it takes a long time until they will be published. But yes, I think this one of the letters is interesting because it's already online.
Yeah, we've had like quite a range of users. A lot of the expected users, I expected a lot of basic neuroscience as you were just talking about. But we've had a huge range as well in robotics, in cybersecurity, of course, in AI, that have come on and are really making excellent use of the platform. For example, our first customer, and they were public about it, so I can talk about it, but was Cardano, the cryptocurrency group, who are really doing some cool work exploring a range of things that I won't talk about. But other things have surprised us too. Like one of the early users was doing cybersecurity. Again, I'll talk about it because there was a publication on it. And that was not an idea that we would have come up with on our own. And I think this shows the value of opening this up to the world, is that you're going to get people who have different perspectives than you and different approaches, and will be able to develop things in ways that you wouldn't. And that's the benefit of democratizing technology.
Yeah. One thing I ran into when I was researching this is that it's very hard for me to, maybe not for anyone else here, but for me it was very hard to understand how you actually program this system, because it's not a silicon-based system in a way. So maybe, Brett, if you can explain with the Pong demo, how do you actually get the neuron to output what you want it to output?
So the approach that we use, just one of many approaches for the Pong work, was leveraging something called the free energy principle. This is a neurocomputational theory proposed out of University College London by Professor Carl Frisson, has since been worked on by hundreds, I think, of academics at this point. And what it basically said is that any neural system will try to predict its environment. And so if you think about it a little more abstractly, if I reach for a drink bottle here, I don't really see the drink bottle in any real sense. What happens is my brain takes in sensory information, it computes a model of the world, and I act upon that model. Now, if I correctly reach for and pick up said drink bottle, my expectation of the world has matched my prediction. This is called sort of means that I've met my expectation. But if I miss it, if I knock it over, then I'll be what's called surprised, technically. Essentially, it's an increase in the information entropy, which means the variability of the model. So this is quite a comprehensive theory. To cut it right down to oversimplification, we said, well, if the neural systems at all scales, right, individual neurons, groups of neurons, whole brains, are trying to predict their environment at different levels, what if we just made them getting an undesirable action, like missing the pong ball, something that can't be predicted, random noise, essentially? Would the cells reorganize their activity to get better at hitting the ball? And that's indeed what we found, that they would actually use the amount of information entropy in the signal as a conditioning agent to modulate their activity. Having said that, I will add a preface. This is just one approach. We explore a lot of different neurocomputational approaches, some of which we'll be talking more about soon. And I think there's huge depth to be explored here. Because if you can understand at that level of information physics what drives intelligence, that's how you really unlock the ability for these cells to learn.
Yeah. And how many neurons are in the CL1?
But for the CL1, it can range. We typically use about 200,000 now. In our early work, we did 800,000 to a million. We can grow several million in one of the MEA, but we find it's not as necessary. You can get better performance for, say, the 200,000 when you tightly control their structure and connectivity. Otherwise, you can get to the case where adding more and more cells means you get more noise relative to signal. So it can be a bit counterintuitive, but again, if you think about it sort of outside of this, a bee gets around its environment very well with a few 100,000 neurons. An elephant gets around its environment very well with more neurons than a human. One doesn't really get down better. It depends what it's designed for and how it works. And so if you design it in the right way, you can get a lot of benefits out of it without having to just think bigger is better.
Yeah. Ewelina, what is some of the bottlenecks that you experience today of working with these organoid technologies?
Yes, I think what you mentioned before about programming neurons is the biggest. I see this as the biggest challenge currently because nobody knows how neurons encode information. So we know quite a lot how they process signals, but we do not know what does it mean. And that also applies for the human brain. We can read a lot of signals from the brain, but we do not know what does it mean. So you cannot read the mind today. But I'm sure that it will be possible very soon. And we, of course, want to do this only in vitro, in the cells on the dish. But yes, that's the biggest challenge, I would say. And also that comes with another question. If you know what does it mean, the signals between neurons, then you know maybe how to change it, how to change connections between neurons. So as I mentioned, the learning in vitro, that's the biggest challenge currently. There are some small progress in this, but it's still small. So we would like to be able to master this. And I would say this is, I see this as the biggest challenge. And there are a lot of other things also related to biology, as also Brett mentioned, like how you design this. This is also a big question because there are many ideas you can have about how you combine different type of neurons, how you do this. And is it better, which structure, which composition is better for computing? So this is actually gives you a lot of options to test. So that's also it's a challenge. Also the biology part, but I would say the data part is the hardest. Understanding the signal and learning how to program.
And
One of the really nice features that we've been exploring with the CL1, because we've got that really tight closed loop, is you can actually break down the waveforms. So I think anyone who's done basic biology knows neurons have these typical action potential waveforms. But most of the time when you're doing work, because most systems can't run at the same speed, they can only look at individual spike times. And so that's the way I like to phrase that is like, if you look at individual spike times, which nearly everyone, including us historically, has done, you're kind of getting the rhythm of music. But if you want to hear the melody, the notes, you need to be able to look at the waveforms because they differ between cells, between types, and in a whole bunch of ways. And so like one thing we've been doing to try to understand it better is actually pulling out in real time these different waveforms. And that's giving us a lot more capability to understand how these systems are working.
Yes, I agree with you. We also do this, and I think it's also interesting to look not only on the spike events. Yes, that is much more rich information, the shape of the spike. Mm.
I think there's also some interesting practical limitations to biological computing. Maybe Brett at Cortical Labs, you might run into it more because you're actually setting these units. I think now the neurons last maybe 100 days or a bit more, but what happens after that? And how do you create a product that you can actually sell and commercialize at a larger scale?
So we don't sell CL1 units with cells in them. If you're buying a unit, the assumption is you grow your, well, the explicit requirement is you're growing your own cells. And so we have minimum lab requirements for our customers and so forth and so forth. For our own purposes, we grow people's cells if for the cloud use or for CROs. And for those, there's quite a range that, as I was saying, cells can last, with some going well over a year. And other types of cultures sometimes are much more short-lived. But again, it's the right tool for the right job. Some of those short-lived ones show some very interesting properties very quickly. So if you just want to iterate really quickly, those are ideal. Or if you want to look at really long-term, you take another approach. So that's the kind of beauty of it, is that there's a lot of modularity and flexibility in how you approach this problem. It really is a living material to be used as you require.
Yeah. I think we already had a question in the chat, which I also wrote down, and it kind of links into this, which is regarding performance and how you measure your system. We already said more neurons is not necessarily better in these systems. So how do you measure performance, and what kind of benchmarks do you look at? And maybe Ewelina, you want to answer that one first?
I think that at this stage is a little bit too early to talk about benchmarks because today the question is how to master, how to have control over neurons. So any type of controls. For example, we manage to store 1 bit of information, so this is very little. So you cannot really compare this at this stage of the development. You cannot really compare this to, for example, digital systems which are ready. So I think it's a bit too early to talk about performance. Definitely, because today the objective is just to have any results which are kind of like you can show that you were able to change something in the signaling of the neurons because of a stimulation. Because many times it's just it seems to be random, and it might look like a totally random response. Like actually, neurons are active all the time. It's called a spontaneous activity. So actually, what you want to show is that what you measure for them is not totally spontaneous, that you somehow affected them intentionally with your signals. For example, you wanted increase the activity or decrease. So very simple things, I think way too early for benchmarking. That's what I would believe. Yes.
But I don't think it's too early to start looking at benchmarks generally. There are really clear-cut benchmarks, closed-loop latency times, reliability metrics of the platform, that can be benchmarked, like the closed-loop latency we showed as one example, but there's dozens of others for our system that can be done. And for the neural performance, I think there's some really clear things too. You can look at the complexity of how signals are processed by the cells. For example, I'll throw this preprint in the chat. It's just a theoretical one, but it does provide a method to look at how information processing can occur inside neural systems that we've got under peer review at the moment. Should be out soon. There's other properties like functional connectivity, criticality, complexity metrics, processing of information entropy. You can also just look at gameplay statistics. So you could look at things like for handwritten digit tasks, what's the accuracy when controlled properly? For Pong tasks, what's the hit-miss ratio? There's a huge number of things that we can start to look at. And I think by standardizing some of these benchmarks, you're going to get a lot better performance in the whole field. Because at the moment, everyone's kind of playing their own game. One person will do things on some sort of chaotic time series prediction. Another one will do a video game like us, right? Video games. But how do you compare those two, right? How do you know which system's working better? So I think now is the perfect time for us to, as a field, sort of come together and start having some consistency in benchmarks, so we're not always talking across purposes. And I think there's some really good ways, like I've just suggested, that we could start being more consistent as a field.
So let's start looking a bit more towards the future. I think there's still a lot of promises, and I think that's also what makes it so exciting. When you're working at your companies, what is the main promise that you keep in the back of your minds and that you're working towards? Just start, Ewelina, yeah.
So the biggest promise is energy efficient computing. So what we want to ultimate goal, what we want to make is a bio server, which will be a remote available computer, which will be computing power actually, which will be much cheaper than digital, because it will use 1 million times less energy than silicon. And we believe that it will make a huge difference, especially in 10 years when it will be ready, we estimate, because of this increasing price and increasing costs of AI, of digital AI.
Are you seeing any AI players already interested in the technology?
Is it to me question? Sorry, can you
Yes,
repeat?
yeah. So continuing on that. So the energy promise is obviously quite relevant in AI. Do you already see AI companies reaching out and interested in the technology?
Yes, I wouldn't say always AI companies. I would say people who are using AI definitely are
Yeah.
interested, or people who have some kind of interest in saving the energy. Definitely, yes, yes. This is big argument. Yes, and many people actually are impressed, and then they believe in the project because actually that's kind of foundation. Why would you bother, to work with totally new substrate? It must be has some advantages. So kind of the biggest advantage currently, which we see, is the energy efficiency. Although along the way we might discover different ways, different properties, because maybe we will be able to develop some superior algorithm. But this is still hypothesis. Something what we know for sure is that it can be energy efficient.
And Brett, did you have any other findings?
I fully agree. Energy efficiency is a very attractive benefit. I think there's some nuance there in that I think like there are cases where we may resolve some of the energy concerns sort of in connected areas. But if you're looking at edge computing, or especially in areas where energy may be limited, then biology becomes incredibly powerful. But I think there's a lot more to biology also than just energy efficiency. And we waste, as much as, I might roll my eyes at some things. Elon Musk said the other day, we waste most of the energy that comes onto this planet. And he's right, of course, we do. So we could get better at that. So I think what's really interesting there is actually the abilities that biology show. And despite the advancements we've seen in AI systems, they don't show general intelligence. They don't show features like intuition and things that we haven't even explored that well. So there's a lot of features there, and we are really interested in the long-term goal of heterogeneous compute. Because at the end of the day, much as like a CPU is aided by a GPU, we want to know what happens when you combine this with the biology, when you start to build out capabilities that have never existed before. And that's why we're so interested in these bioengineered reproducible approaches. Because the focus isn't just how do we do what we're doing now, but like more efficient. It's how do we get to things that are not possible otherwise. And the nice thing about this is if you look at AI, it's really unknown with what you can get there, right? What we've built now is the best silicon thinking, in quotes, system we've seen. But biology, we know it has some amazing properties, not just in humans, but consider what bees do or octopus. There's a huge amount of capability to be explored in terms of how it processes information. The question becomes, how do you get there? And that's a pretty exciting space to work in.
Yeah. I think some people dream of biocomputing one day being part of the infrastructure of larger systems, but can it really be reliable enough to work on that scale as a computing infrastructure?
I think so. We've achieved some things that do show quite a lot of reliability, but it's a trade-off, right? Humans, famously unreliable in a lot of ways, we can still do a lot of amazing things. So you could build more and more reliable biological systems. Consider the biological, or let's say the cognitive, for the given quotations again of that word, capability of an individual bee. Not a huge amount of variance, but collectively they work quite well together. Compared to a human, significant variance, more capability. So it may very well be that there is this trade-off, or it may be that there's ways to remove that trade-off. But again, I come down to this, the right tool for the right job. What are we building? Why are we building it? And then how do we get there?
Yeah. Do you
Yes,
want
I
to add
fully agree.
anything?
Yeah, I think a human brain is great kind of proof that neurons can be reliable for some tasks. Yes.
I think before heading into the Q&A, because there's quite a lot of questions, I have one question which I really wonder, and that is, whose cells are in these systems? Is it yours?
Everyone is asking for that, but this is solved. Really, that's not a controversial topic as it seems to be for some people. So we are using, I think both of us, but Brett can also clarify. We use neurons which are derived from human skin. So actually, stem cells derived from human skin are commercially available, so you can just buy them. Like you buy any type of other chemicals or equipment to the lab, you can buy also different type of cells, including also this. So that means that all the process of ethical consent and everything is solved, and these cells can be used for research, for any type of research, of course. So yes, so this is commercially available, and we from them we produce. We have our own expertise also in the lab. We produce in Switzerland always our own neurons from that starting stem cells of human skin. And this is why actually we can produce huge amounts of these cells without any sacrifices. So we don't use animal cells or anything like that. So that also makes actually such a project possible, because in the past it wouldn't be possible to work on biocomputer, because we would have to sacrifice animals and it wouldn't be ethical to sacrifice them. But here it's just a skin. Yes.
And Brett, is it true that your CEO Han did integrate his neurons into one of the computers?
Yeah, our donors' identity are confidential through the ethics approval we have, but Han has publicly stated this before, so I guess he's waived that confidentiality. But it's really just proof of principle that this is as you know, as you're just hearing, this is a virtually harmless, same as giving blood to a doctor for a test, very routine. Or you could, someone the other day made iPSCs out of urine. So it's not like it's some special magical thing. It's simply taking cells, converting them back into their pluripotent state, and then you choose a direction. And beyond just the ethics, it also gives you so much specificity. You can engineer different types of cells, as we've been doing, and really build more and more complex networks. Again, to try and build you the right tool for the right job, use all of the breadth that biology, that evolution has given us. And maybe one day also explore going beyond that. There's a lot of opportunities out there, and we're just scratching the surface.
Fantastic. Yeah, for the sake of time and to keep it a bit interactive, I want to give the word to Jemima, who can relay some of the questions from the audience.
Yeah. I think, has Brett's computer frozen? Oh no, it hasn't. Sorry.
Yeah.
My bad. On the topic you were just talking about, I wanted to ask a question about, like a personal question about kind of, there's obviously organoid intelligence is being seen like to help understand our understanding of brain development and learning and memory, and kind of actually potentially with treatments for neurological disorders. And is this the kind of thread line that you guys are looking into at the moment, or could it be a future line of your research, or would it be something separate?
I can jump in. Yes, we are looking at that. We work with a number of different researchers and some companies who are looking at that. So I think it's an incredibly exciting opportunity. And one of the papers we published actually showed that this was looking at a simplified epilepsy model. And we actually found that when you had just the cells, you gave them drugs without being in a closed loop environment, you got a very flat response across all of the different doses and all of the different anti-seizure medications. But when they're in a closed loop environment, suddenly differences started to appear. And I think this sort of embodiment is so important for neural cell cultures, because when you, again, to think about it, neural cells did not evolve to live in an information void. If you take a person and you put them in solitary or one of those sensory deprivation chambers, weird things start happening. And that's because neural systems evolved to be in an information-rich environment. And so testing drugs when cells are in an information void, as we've done for all of the in vitro testing that's existed, pretty much, is obviously not the best approach if you want to meet physiological relevance. So I think this is a great direction forward.
Thank you. And then from the chat, there's a question here about whether excitotoxicity is ever a concern when you're kind of dealing with these custom cell structures.
What do you mean?
You want to go on that one, Ewelina?
I'm not sure which kind of toxicity. Can you repeat?
Excito.
Oh my God. Yes. Well, so actually, I would say neurons are fragile, let's say that, and many things can happen. And actually, we started the lab as engineers. So the founders of FinalSpark are engineers. So of course, we started sometime. But what is advantage when you have living cells on MEA, on electrodes, is that you immediately see if something is wrong. You can even see if you open incubator door, which is actually not considered to be toxic for neurons, but you even see this on the data, because we have real-time 24 hours per day, 7 days a week monitoring. You can see examples on our website. So yes, of course, some kind of toxicity can happen, and cells might die, and they dying, all the time, because we have many systems, and we just replace them. Yes, but let's say that for us it's important that we keep them healthy, that we keep them alive, and if something is wrong, we exchange them. Yeah. But we don't study the mechanism or something, if that was the question. But you could theoretically, yes. And what is also good, we haven't done this, but theoretically for our users, is that you can use the platform to study any type of toxicity, also chemical, because you immediately see the results on the electrodes. So that's also the advantage. Although this is not the purpose of the project, but we still use the techniques of electrophysiology, so we can do a lot of stuff which neuroscientists might be interested in, which maybe are not necessary for biocomputing.
Mm-hmm.
So you can do a test of toxic substance, for example, to keep it simple. Yeah.
Thank you. There are kind of a few questions around, I guess, the fact that we're dealing with living systems and kind of replicability. So if you're able to create cell cultures that are then kind of reproducible, and also when looking at what does version control look like for the biological state? And then if a culture learns something's over 2 weeks, can you kind of snapshot this and restore it?
I love that question because that's such a natural thing from silicon systems. You just save it
Yes.
and you load the file again years later, it's
Yeah.
still there. But that's not like how biology works. Now, you can have very reproducible systems, and the systems can learn and reorganize functionally very quickly, minutes, not hours or days. And I'm not saying that we'll never get to the point where we could indeed do this snapshot, but the implications of snapshotting effectively learning and memory in neural systems, that's almost getting to the, if anyone remembers the Matrix, the I Know Kung Fu scene, where they just download or upload Kung Fu to Neo. That's kind of what we're talking about here. We're talking about writing, learning, and memory into neural cultures. I don't know if that's even possible. And maybe if it is, maybe these systems can help us get there. But I think that's still a very long way off. But I think, again, that's the exciting promise of this technology is we don't know what's out there. We don't fully know what's possible. All we can commit to is exploring it rigorously, patiently, and opening that exploration up to everyone who's interested as much as we can so that we can try and accelerate that exploration together. Because there are a lot of potential benefits, not just from a computing perspective, but potentially how it reflects to us, and for that matter, every other neural system in the world.
Mm-hmm. Thank you. And then just in the interest of time, because we've got 2 minutes, I think, I don't know what you think, Dom, but if I kind of think of ending with the ethical question here. And where do you think we should draw the line as these systems become advanced, and especially making them remain safe and meaningful and under human control? Or should we even worry about keeping it under human control?
So actually, these are very important questions. Every new technology is bringing,
Mhm.
and we put a lot of effort to reach out to philosophers to help us, because we are just scientists and engineers. So not, of course, we have our opinions, but experts from ethics are more philosophers, and there are many people actually who work on the philosophy of technology, and we hope they can help us to answer all these questions. I can only say that for sure, when you create new technology, you have to monitor this, because you cannot really, you don't know how it will end up. Of course, we have good intention, but we just have to monitor what will happen next, and it's important that philosophers study also every new technology and think about this and help us.
Hmm.
Yeah, I fully agree. And it's so important to engage with the broader community. We've been really focused on that since Cortical Labs started. In fact, our first scientific paper as Cortical Labs was an ethics paper, not the Pong paper everybody knows us for. Uh, I've actually put 2 links in the chat. One is a very recent large consortium of ethicists that we worked with on organoids and using organoids for a range of purposes, including this. And the other is a short article in Journal of Neuroscience that I took the lead on, where we basically proposed what, actually I didn't come up with the term, that was Professor Lomax Boyd over at Johns Hopkins University, but what he termed experimental neuroethics. And essentially it's allowing us to explore as a form of anticipatory governance, the ways to approach this research ethically and sustainably, and actually be able to look for what people call morally relevant states as you go. And we've been really taking that on in a serious way, and it generates such interesting work and discussions. And I think it also is important for people to realize that the scientists and industry engaged in this, and I've talked with Fred over at FinalSpark and other people at FinalSpark, and we're all on the same page. It's so important for people to know that this is not the case of sort of science gone amok. This is actually something with people who care about the technology, who are building it because we believe it can be ethically good, and we're putting the work in to make sure that's the case. It's not just hope for the best. You can actually choose how to develop your technology, as weird as that sounds.
Yeah, you kind of embed it in there.
Exactly. That embedded ethics is actually the formal term, and that was core of the organoid intelligence work as well that you talked about. I was part of that as one of the sort of founding scientists of the movement, and embedded ethics was central to it as part of that initial thing.